Hey everybody, in this lecture I'm going to talk about the environmental hazards to human health. And we'll look at a lot of case studies and break down sort of what are the biggest risks to human health and how do we assess risk from those different conditions. As we go through the next few units, we're going to talk about some of these risks, specifically those that come from poor air quality, those that come from poor water quality, those that come from exposure to toxins from solid and hazardous wastes. So to assess major health risks, we want to know what the risk is. What is the probability of suffering harm from a hazard that results in either injury, disease, death, damage, etc. And when environmental risk assessors do this sort of thing, they look at both the likelihood of exposure and after the likelihood of exposure, what is the likelihood that that exposure leads to harm? So we want to know, okay, what is the exposure? What dose do you receive of a particular chemical? How often do you interact with a particular animal? How often do you eat a particular food, etc.? So that's going to look at both dose and frequency of exposure. But the conditions around the person exposed also can contribute to their risk. If you are older, you may be at a higher risk. If you are younger, you may be at a higher risk. It really depends on the age of the person, the health of the person, the sex of the person, and their general genetic conditions before exposure to that particular risk. So risk assessment is using statistics to basically estimate how much harm a particular hazard is going to cause. And if the risk is high enough, and if the risk is high enough to enough people, we can enact management plans to be able to deal with specific risks. So what is the hazard? What is the likeliness of the event? And what are the consequences? What is the outcome of that particular event? So under risk management, how does it compare to other risks? We may say, okay, that's a risky situation, but that person is less likely to be exposed to it, so we're not going to worry about it as much. Or we may want to know, what is the risk reduction? Do we need to reduce risk? If you are, exposed to toxins in your water supply repeatedly, you're going to need to work on a plan to reduce that risk. How will it be reduced and how much money should be spent kind of depends on who's at risk and sort of the size of the population that may be at risk from a particular hazard. I always tell my students, if you want to make a lot of money, go into risk management. Insurance companies are always looking for this kind of information. And they are looking for people to be valuable and accurate risk assessors so that they can determine how to best protect their clients. We're going to go through 4 types of hazards. Those include biological pathogens, so bacteria, viruses, parasites, etc. And I'll try to give you an example of 3 of those. Chemical hazards, so those can be chemicals that we are exposed to in the air, in our water, in our soil, in our food, etc. Natural hazards, which we've talked somewhat about throughout the year, but those include things like risk to fires, storms, floods, etc. Insurance assessors are always trying to basically determine the mathematical risk associated with those things. And then some risks are sort of cultural based or lifestyle based. Things like car accidents, criminal activity may be cultural based on where you are located. Risks that come from your lifestyle may be determined by your diet, your likelihood to smoke, etc., etc. So let's start with biological hazards. An infectious disease is a pathogen such as a bacteria or virus that invades the body and multiplies. And we're most concerned about biological risks that are transmissible, in other words, can be transmitted from one person to another. But there's plenty of diseases that are non-transmissible, not associated with what's considered a pathogen. They do offer a lot of risk to people, heart disease, cancer, etc. But we don't have to track them as much in terms of exposure because they aren't transmissible. The deaths per year in the world are most uh, from a disease, I should say, are most going to be those of lower respiratory infections, bacteria and viruses that attack the respiratory system. This can include influenza and the flu. This would include COVID-19. And so these are serious conditions. Tuberculosis is still a huge one. We'll talk about that. HIV and AIDS, major virus. We'll talk about that. Hepatitis, diarrheal diseases, malaria, we will talk about that, and measles are some of the biggest biological hazards that we see transmitted today. And you guys are all familiar with the terms epidemic and pandemic. When we have a wide-scale outbreak, we refer to it as an epidemic, and a bunch of, policies will start to go into place to prevent the spread. Obviously, if that spread gets wide enough, we enter into what we call a pandemic, which is what we're in right now. Unsurprisingly, our textbook, which is pre-COVID, talks about emergent diseases on the rise. And they talk about sort of what are the reasons for that. Resistance to antibiotics, disease-transmitting species, developing resistance to pesticides, zoonotic diseases that jump from animal to human, etc. So COVID-19 would fall under this category of a new emergent disease. So let's look at some case studies. The three big ones that your textbook would cover include tuberculosis. This is a bacterial infection that affects around 9 million people per year. It kills about 1.7 million people, so its virulence is pretty high. We are seeing more increases in tuberculosis today, mostly due to lack of screening, lack of control programs in countries where TB is highest. But the biggest problem associated with tuberculosis is that the bacteria itself is becoming resistant to our antibiotics. We are seeing antibiotic-resistant tuberculosis, and these strains of TB are going to be more deadly because we can't fight them. So that's sort of what's going on with TB. HIV and AIDS is sort of the big case study when we look at viruses around the world. In 2014, there were 36.9 million people infected worldwide with AIDS. About 1.2 million of those are here in the United States. According to the World Health Organization, we see about 2.2 million new cases per year. And this is really troubling because despite the incredible amount of research into HIV and AIDS, we still don't have a vaccine for HIV. And generally speaking, if you get AIDS, you are likely to eventually die from it, although new drugs have come up, have been researched, have been put on the market to help infected people live longer. Unfortunately, the access to those medications is limited, and where we see HIV and AIDS tends to be in nations that are still going through their demographic transition, and most of the public can't afford the drugs that are going to be able to fight HIV and AIDS. And so we've seen life expectancy, particularly in sub-Saharan Africa, reduced from 62 to 46 years. And life expectancy has actually dropped 40 years in the 7 countries that are most severely affected by AIDS. This is not surprising. We saw some of this data when we did the demographics lab back in the fall. So it's just very disturbing that the second biggest killer virus globally, and the leading cause of death for people ages 15 to 49 still doesn't have a vaccine to be able to prevent it. How can we prevent it if we don't have a vaccine? We need to reduce the number of new infections to prevent further spread. This is going to mean concentrating our efforts in groups where the disease is likely to spread the most, providing free testing, and educating people to go get tested when they can, providing free or low-cost drugs to slow the disease progression, providing contraceptives to be able to prevent the spread of HIV through sexual transmission. There's a lot of priorities that we need to put to place to be able to prevent the spread of HIV and AIDS. And the World Health Organization, UNESCO, UN, etc., are all working on those initiatives, but we need sort of a bigger push, and we really need to start working on or finding our vaccine for this virus. Probably one of the biggest risks to people in the tropical regions of the world is malaria, which is caused by a parasite called Plasmodium. Plasmodium is known to basically develop early stages of its life in still waters, where mosquito larvae will pick it up. As the mosquito larvae pick it up, they go through their metamorphosis into their adult stage, and those mosquitoes will inject the Plasmodium into a human host. The early life stages of that Plasmodium will actually enter into our liver system and develop its next life stages, which then can be transmitted through mosquitoes into the water again when they go to lay their eggs. So there's sort of this life cycle that involves still water, mosquito exposure, and consequences within the human body itself. We have spent billions worldwide on malaria treatment, and we've seen some positive effects around this. 63% drop in malaria deaths worldwide since 2000, which I think is just a remarkable project. And a lot of that has come with the expense of fighting the mosquito. DDT, even though it is banned here in the United States, is being used to combat mosquitoes in some areas. Mosquito nets have been highly promoted and provided for free to people in the biggest at-risk locations. And the use of a mosquito net can greatly increase your resistance to be able to get malaria. So, organizations like the World Health Organization work on reporting global death rates for infectious diseases. They are projecting these things to decrease, continue to decrease because of our access to technologies. But as we see with climate change, etc., it's becoming harder and harder to fight infectious diseases. So vaccinations are one of the most important solutions to infectious disease. Vaccinations have increased for things like tetanus, measles, diphtheria, typhoid, polio, etc. Since the early 20th century. We are starting to see some anti-vax or vaccine resistance from the general public, which is alarming to a lot of public health organizers and infectious disease experts, because they are greatly concerned that populations that had at one time eradicated some of these diseases are going to start seeing them again. Measles is a really good example of that here in the United States. And then one of the other biggest things, and we'll come back to this when we talk about water quality, is that oral rehydration therapy has been able to combat dehydration due to diseases that result in diarrhea. So something like diphtheria or cholera that's going to normally seriously kill off a number of people in areas where you see them. We are doing better as the World Health Organization to educate others about how to boil their water, how to include electrolytes, and how to do rehydration therapy so that we can get over these waterborne diarrheal diseases that can kill, and especially can kill children. And if you think back to the population lab back in the fall, we talked about infant mortality rates and how often infant mortality is correlated with poor access to quality drinking water. So beyond biological hazards, we have chemical hazards. Toxic chemical is one that causes temporary or permanent harm and can cause death in some cases to humans or animals. The 5 top toxins here in the United States, according to the EPA, are arsenic, lead, mercury, vinyl chloride, and polychlorinated biphenyls, or PCBs. And so there are three main categories of chemical hazards. Carcinogens, which is a chemical or radiation or a virus that can cause cancer. So the chemical ones would be the ones that we're looking at here. And things like arsenic, formaldehyde, PCBs, certain chemicals in tobacco smoke, etc., would all fall under that category. A mutagen is a chemical or a radiation that causes some sort of mutation in the DNA that may or may not lead to cancer. And then teratogens are chemicals that cause harm by basically inducing birth defects during embryonic and fetal development. And so these are often going to be the chemicals that the EPA, the FDA, etc., will warn pregnant women to avoid during pregnancy. And even something as, normally we don't really consider it a huge hazard like caffeine, is going to be a type of chemical that is cautioned against for pregnant women during different stages of development for the embryo or the fetus. PCBs are maybe a term that you're not familiar with, but this is a class of chlorine-containing organic chemicals that are really stable in the environment. If we look at the chemical structure up here, we see that we've got a double ring structure with double bonds within that ring. That double bond, double ring structure is super stable in the environment. There are almost no known natural enzymes that can break this molecule. And it's used in a lot of places. It's used in lubricants, hydraulic fluids. It is an insulator in these electrical transformers that you see at the top of power poles. Because it's very stable and non-reactive, it's known to be used in fire retardants for things like paints and fabrics and electronics. So it does a lot of good in the products that we use it in, but if it gets into the atmosphere, it dissolves into water. And because there are no real natural enzymes to be able to break it down, it's biodegrading at a really slow rate. And it is known to bioaccumulate up through ecosystems, so that if you have a small exposure of PCBs in your water, the sediment's going to take some of that up, your plankton and phyto and zooplankton are going to take some of that up, the fishes that eat those smaller organisms take it up, and it sort of bioaccumulates up the food chain. I have a close friend who researched PCBs from a plant near her home in Massachusetts, and was able to actually document the transfer of PCBs from the water to the algae, to the snails, to the fish, to the fish that are edible, and sort of big fisheries in the area. So a lot of PCBs were banned from use in the 1970s, but we still see them in air and water and soil and organisms, and there are some products here in the United States that still have and contain and can use PCBs. BPA is another big chemical that a lot of people hear about. Bisphenol A is an estrogen mimic molecule, a chemical component in a lot of hardened plastics. And over the last, I would say, 10 to 15 years, studies have come out and some determined BPA is problematic because it can disrupt fetal development. Because it's a hormone mimic, it can lead to feminization in males. It can lead to early puberty onset. There was a lot of potential hazards associated with this chemical. In more recent years, other studies came out indicating the risks were a little bit lower. The FDA says that BPA in food and drink containers is not high enough to be a health hazard. And 2015, the European Food Safety Authority kind of agreed with that, saying we just don't see levels high enough in humans. But there is plenty of evidence that suggests that if this material is in a plastic, it can leach into the food or the drink that plastic is containing. And we especially see that leaching happen when these containers are heated if the containers contain an acidic material like a juice. So the EPA has set threshold levels for the amount that can be found in the body, and it's up to the companies to make sure that their material doesn't leach into their food, which then gets into the body of their consumer. And typically we examine the BPAs coming out of urine levels. One of the biggest questions around BPA was exposure in children and infants, because a lot of children's toys are made out of BPA-based plastics, and baby bottles, which are again BPA-based plastics, in some cases were being heated up to warm the milk before getting it to the infant. So there's been sort of a big move or a big push to ban materials that contain BPA, and companies on the whole have pretty much complied with that because of the public perception of what's going on with BPA. They replaced it with bisphenol S, but newer studies are indicating that BPS may have similar effects to BPA, and we're starting to see it found in the urine of people that we tested for. So we may have just moved from one plastic material to another, and we may end up with some of the same conditions or the same situations. So when we look at a chemical, we want to determine how harmful that chemical is to somebody who's exposed to it. This is what we call toxicology, the study of a harmful chemical and the measure of the response in individuals to that chemical. Is their exposure acute, which means just sort of one example of an exposure, or is their exposure chronic? They repeatedly get exposed to this particular chemical, it builds up over time, and it causes issues throughout their life. So we want to measure the harmfulness of the substance, and the harmfulness of the substance can depend on a lot of conditions, and it can change from person to person. So what is the dose? What is the amount of chemical that's been exposed? Has the person ingested it? Have they inhaled it? Have they somehow absorbed it through the skin? We want to know what is their dose and how did they get it. We are going to question the age of the person exposed, because if you're very young or very old, you're likely to experience negative effects or have higher health hazards as associated with a chemical than if you are young, middle-aged, or healthy. What is that person's genetic makeup? Do they have a natural genetic predisposition to to a toxin that makes them more sensitive to that particular chemical? And we also care about that level of chemical in the environment, because the toxicity can also be applied to animals, to plants, etc. So is that particular chemical soluble, which means it can be transmitted through water? Does it have some sort of persistence? So things like BPA, DDT, etc., with these really beautiful carbon ring structures with double bonds in them, are very, very persistent. And what is the likelihood that they biomagnify in the body or bioaccumulate up the food chain? So those are two things that we're going to think about when we look at toxicity. We'll talk about what we call LD50 in a later assignment, but essentially a dose-response curve is going to be used to determine, usually via test organisms, what is the median lethal dose, the LD50, basically the dose that kills 50% of the test population. Obviously, we're not going to do LD50 assays with humans because that is unethical, but we do use vertebrates, we do use mammals, and we try even to use organisms that are closely related to humans. So I've seen, primates used for LD50 studies. I've seen rabbits used for LD50 studies, etc. But essentially, if you hit a specific dosage that causes 50% mortality, that's the LD50 for a particular toxin. And like I said, we'll talk more about this in a later assignment, but it's something important to remember. We have a series of potential impacts from these toxins and how they have an impact on the body. We can have chemicals that compromise the ability's immune system. So things like arsenic, methylmercury, dioxins are going to attack the immune system, prevent a person from being able to fight off other pathogens and make them more susceptible. Neurotoxins affect the nervous system and can lead to behavioral changes, disabilities, paralysis, death, etc. And we often see neurotoxins associated with pesticides. But PCBs and methylmercury would also be considered neurotoxin. Lead is a good example of a neurotoxin, etc. And then like BPA, we have these sort of chemicals that mimic or have a similar shape to hormones, have a similar shape to steroids, and are going to have an impact on the human endocrine system. We usually call these hormonally active agents. So in a normal cell, you have a hormone that meets a receptor on its cell to induce its, whatever its metabolic pathway is. If you have an estrogen-like chemical, something like BPA, if it has a similar shape, it's going to bind to that receptor. It may block the body's ability to normally get its hormone, or it can mimic that hormone and induce whatever that hormone's sort of pathway is going to be in the body. So those are biological risks and those are chemical risks. When you go to assess the risk to a specific group, there are variations in the sensitivity of a toxic chemical to a pathogen really within a population, mostly due to genetic variation, but also due to age. So children are the most susceptible to toxic substances. And there's a few reasons why that is. One, when you're younger, you basically are going to breathe more air, drink more water, and eat more food per unit body weight than adults are. Kids are growing, they have a high metabolism, they're constantly eating, they're constantly drinking, they're constantly having a high rate of breath comparatively to older people. Kids are going to be more likely to be exposed to toxins because they don't know not to touch. They are going to touch things. They're going to put fingers in their mouths after they've touched things. They're going to eat lead paint chips. Kids generally are going to be at a higher risk simply because behaviorally they don't know any better. When you're young, you have a less well-developed immune system, and your liver is smaller and it's not going to be able to detoxify in the same way that adults are. So, when we think about children, they're definitely going to be in really high risk groups. So risk assessment in, children's clothing, children's toys, children's playground equipment, etc. All of that stuff is going to be a little bit different than the similar types of risk assessment you would do for adults. So estimating risk from using basically we estimate risks using a lot of different technologies, but it's hard because we can't necessarily predict human behavior. We can't predict chance of risk as easily as we would like to, and we certainly can't predict sabotage. So that's kind of a problem when we talk about risk assessment. The reliability of a system is multiplicative. So what does this mean? You have to think about both the risk of the technology or the material or the pathogen, etc. itself, and then multiply that by the human reliability to operate that technology, to reduce their own exposure to that chemical by using it correctly according to manufacturing guidelines, etc. And the example given in your book is about nuclear power plants. So if it is a 95% reliable plant where we have a low risk of something going wrong, but human reliability is only 75% because there is always a risk for human error, then the overall reliability is going to be 95 by 75, which is about 71%. So systems reliability is synergistic, I would say, where we have to think about both the system as it's designed and also the human who's actually operating that particular system. The same could be said for use of exercise equipment at home, use of a vehicle, um, the likelihood of risk from walking across a bridge or something of that sort. So when it comes to risk analysis, we have determined that poverty is the greatest risk to health worldwide. And why is that the case? Because if you are living in poverty, you may be in a malnutrition state. You may be exposed to disease. And the reasons for that is you don't have adequate sanitation facilities. You don't have enough fuel to both heat your home, to keep you warm, and to do your cooking. So you may have exposure to pathogens in your foods. You may have exposure to cold, which could lower immune system fighting, etc. And the same could be true for access to electricity, healthcare, housing, drinking water, etc. So poverty, as we have been able to determine it in our risk analysis, is the greatest risk to health worldwide. Air pollution is the second, and then tobacco use is the third, and this is globally, but in the United States, it's the most preventable cause of sickness and death. It's basically been attributed to 48,000 deaths per year. And that's far surpassing even the next closest risk in the United States, which is motor vehicle accidents or other accidents in the home that result in death. If we think about COVID-19 and its impact on the United States population, we are over 500,000 deaths this year due to COVID-19. So, it has surpassed the amount we would typically see from people using tobacco. And I think the CDC just came out and said it was the third highest cause of death this year in the United States. So often on the AP tests, they'll ask sort of how can you promote risk prevention? And the easiest, things that I would say to do that are always to increase access to screenings. That's for exposure, pathogens, non-transmissible diseases, etc. Increase access to preventable measures. So something as simple as a mosquito net is going to go a long way to preventing malaria risk. And education programs. We need to educate the public to engage in risk avoidance behavior if they want to increase their lifespan. And here in the United States, it's not surprising some of the hazards that are associated with shorter lifespans. Obviously, poverty plays a huge role, smoking plays a huge role, but some of these are surprising to students, that being born male shortens your lifespan comparatively to if you're born female. Alcohol and drug use, smoking, driving, interestingly enough, being unmarried is also associated with shorter lifespan. I think this is just a really interesting graph to really think about the risks, and hopefully that will help a lot of students and a lot of people understand that educating about these things, working to avoid things if we can extend life expectancy and reduce risk.